Good afternoon, everyone. I'm very glad to see many of you today. Welcome to my PhD defense. I want to tell you what I did during my PhD project. My PhD project is about how we explore chemicals coming from plants as efflux pump inhibitors against multi-drug resistant bacteria. Let's see this person. So someone is just feeling healthy, he feels perfectly fine, until suddenly he got exposed with bacteria. And this person becomes ill. When someone gets infected with bacteria, they take antimicrobials or antibiotics. And he should be healthy again. However, in some cases, the bacteria are not responsive anymore to the drugs. This is called antimicrobial resistance. Why does antimicrobial resistance become harmful? Because more than 7 million deaths are associated with antimicrobial resistance. And this antimicrobial resistance bacteria can be found in various sectors. Can be found in hospital, in animal farming, in cows, in chickens, and also it can be found in food production environment. What can cause antimicrobial resistance? This tiny microorganism, this bacteria, is very smart. So it has the transporter, which is called the efflux pumps, that help the bacteria to protect themselves by pushing out the antibiotics, making the medicine less effective. So we try to find a way how we can block the activity of these efflux pumps. WHO declares bacteria, Staphylococcus aureus and E. coli, they are categorized as the most critical bacteria. Staphylococcus aureus causes skin infection, and E. coli causes diarrhea. Interestingly, these two bacteria, they are considered as foodborne pathogens. And when we consume these foodborne pathogens, we can also get the food poisoning. Interestingly, plants, when they are treated by stress, so they are in stress conditions, they can produce the chemicals coming from plants. We call also phytochemicals. And there are some phytochemicals from defense mechanism that we are interested in. The first one is the prenylated isoflavonoids, because they have the prenyl group that are put in red circles. And the second one, the indole alkaloids that have the indole ring that are put in the red color. And we want to test the activity of these phytochemicals as efflux pump inhibitors in bacteria, targeting NOR-A efflux pump in Staphylococcus aureus, and also targeting ACRB efflux pump in E. coli. The aim of my PhD research, the first aim, we want to find phytochemicals targeting efflux pump in Staphylococcus aureus and E. coli. And the second aim, we want to understand how the phytochemicals act as efflux pump inhibitors. How do we assess the efflux pump inhibitors? So we did an experiment in the lab by using a chemical reagent, which is called ethidium, and the experiment is called ethidium accumulation assay, and the second experiment is called antibiotic potentiation assay. In our first experiments with ethidium accumulation assay, we have the 96-well plate. Then we add the prenylated isoflavonoids compounds. We also add the chemicals, which is called ethidium bromide. And we need the bacteria that we want to study, the Staphylococcus aureus, that have many efflux pumps inside that bacteria. And we measure the efflux inhibition with a machine, which is called spectrophotometer. What we expect, when the efflux pumps are inhibited, then more ethidium accumulate inside the bacteria, and they can intercalate with DNA, and it will give an increase in fluorescence over the time. Based on my PhD research, we found these two phytochemicals, namely glabrin and neobaba-isoflavone, that show efflux pump inhibition. Well, one experiment is not enough, so we try to do it in another experiment to assess the activity of our compounds. So the second experiment that we did is called antibiotic potentiation assay. We again used the 96 well plates. We have each of glabrin, neobava isoflavone. We also have the antibiotic ciprofloxacin that we want to test, and we also have the bacteria, the Staphylococcus aureus. What we expect, if the efflux pumps are inhibited, then more antibiotic will accumulate inside, and the combination of antibiotics and epi, it will reduce the antibiotics dose. In our study, we found that this compound, prenylated isoflavonoids, can reduce the dose of antibiotic ciprofloxacin by 4 times. What we measure is the concentration to kill the bacteria, which is called MIC. Without the efflux pump inhibitor, the MIC is 8 microgram per milliliter. But over the increasing concentration of prenylated isoflavonoid, we found that the MIC was reduced into 2 microgram per milliliter. So we see the potentiation with the combination with prenylated isoflavonoids. The first finding in my PhD thesis, we found that prenylated isoflavonoids, mainly glabrin and neobava isoflavone, showed efflux pump inhibition activity against NOR-A in Staphylococcus aureus. We come up with the next question. What molecular properties make prenylated isoflavonoids active as efflux pump inhibitors in Staphylococcus aureus bacteria? To answer this question, we use computational approach. We use a software which is called MOI, and the study that we did is called QSAR. So the QSAR method is used to predict the chemical properties that make the compounds active or non-active as a drug. For the QSAR study, we use larger data sets. So we have 37 prenylated isoflavonoid compounds. And we did the same experiment, the ethidium accumulation assay, and we got the information. We called it data set 1. And we also did antibiotic potentiation assay, and we have the second data set. And then with this data set, we built the QSAR model with a software, which is called MOI. The second finding in my thesis, we found that some molecular properties are important for prenylated isoflavonoids to be active as NOR-A efflux pump inhibitors, such as the fractional polar surface area, the hydrophilicity and hydrophobicity balance, so how the compounds, whether they like water or they do not like water, so the balance between these properties affect the activity. And the last one, the overall hydrophobicity, so whether the compound like the lipid more than the water. Then we also have another interest. What about the efflux pump inhibitors against E. coli bacteria? For epi in E. coli bacteria, we are interested in indole alkaloid compounds. So this indole alkaloid coming from Brassica family. So basically, these plants coming from cabbage, mustards, and any other edible plants. And we found this compound, namely camelexin, that show also efflux inhibition in ACRB overexpressing strain. And this compound also potentiates many antibiotics. Based on our antibiotic potentiation assay, camelexin reduced the dose of antibiotic oxacillin by 32 times. Then we come up with the next question: how does camelexin bind to ACRB efflux pump in E. coli bacteria? To answer this question, we use another computational method, which is called molecular docking. Molecular docking is used to see how a small drug molecule fits into a protein, like finding the right key for a lock. So imagine the small molecule is the camelexin and the protein is ACRB. So we try to predict, okay, how camelexin binds to ACRB. So we did a prediction with computational method, and we tried to find the position where camelexin binds. And we found that camelexin binds in ACRB, and the binding area is called distal binding pocket. So we come up with the third finding in my thesis, Camalexin is active as API against ACRB efflux pump in E. coli bacteria and mainly binds in the distal binding pocket area. To sum up what we found in my PhD project, in my PhD thesis, the first finding, we found glabren and neobava isoflavone showed efflux pump inhibition activity against NORA in Staphylococcus aureus. We also tried to understand what kind of molecular properties that are important, and we found the polar surface area, hydrophobicity, and hydrophilicity are linked to the NORA activity of granular isoflavonoids. And the third one, we found an inhibitor for E. coli bacteria, which is called camalexin. And we also find that this compound binds in the distal binding pocket. This PhD project allows me to learn about the potency of phytochemicals. To end my presentation today, I found that phytochemicals offer hope in combating antimicrobial resistance, showing that nature still has answers science is seeking. So this is the agenda of today. After my layman speech, we will have the scientific discussion of my PhD thesis, and it will be followed by PhD committee deliberations and announcement afterwards, and hopefully I can sign the diploma, and we will have dinner and celebration afterwards. And this is the seat layout of today. I have my paranames, Sylvia and Donia, and after this, the opponents will be sitting on my right side, and also my Deputy Rector Magnificus, and my promoter and co-promoter will be sitting on my left side. And I also have my another paraname, Pauline, also sitting in the audience. Thank you for your attention today, and that's my layman speech. So now we are waiting for the committee to come.
Please be seated. I hereby open this ceremony convened by the Academic Board of Wageningen University, in which Marina Irianti is offered the opportunity of defending a thesis with propositions entitled Exploring Phytochemicals as Efflux Pump Inhibitors Against Multidrug-Resistant Bacteria. The defense will take place before an examining committee appointed by the Academic Board as a prerequisite for conferring the degree of doctor. Good afternoon, ladies and gentlemen. I would like to welcome you all to this graduation. My name is Renger Witkamp. I'm Professor of Nutritional Biology and representing the Academic Board and the Rector Magnificus here today. I would like to call the first examiner, who is Professor Wells. Professor Wells is Professor of Host Microbe Interactomics here in Wageningen. Professor Wells, the floor is yours. Welcome.
Thank you. Respected candidate, could you please ask one of your paranames to read out Proposition 5?
Dear paraname, could you please read proposition number 5?
Ignoring both human and microbial cultures reflects a misunderstanding of the foundations of health.
Okay. Your fifth proposition, as we've just heard, uses the word culture in a very interesting way. Could you explain to me what you mean in this proposition by human and microbial cultures?
Yeah. In this proposition, the human culture actually, it belongs to this human socioculture. Because the human socioculture affect the eating behavior, for instance, what type of food that you normally eat. And it's interesting that this eating behavior also affect the gut microbial culture in the end. And we know that this gut microbial culture we also call it a second brain because it affects the overall health. So that's how this proposition comes from, because I want to highlight the important word, the culture, but it has two different meanings. The first one related to the social culture, that relates to the eating behavior culture. And also this eating behavior culture will affect the microbial culture that we have in our gut.
If I understand you correctly, you're saying that our dietary habits might influence the microbial ecosystem and the possibility, or is it the plants that are really having the effect on pathogens?
What we eat, actually, about eating behavior, like about what type of food that we consume, for instance, like the dairy foods or non-dairy food, that will affect the microbial culture that we have in our gut.
Yes, I understand, yeah. But even, well, I'd like to argue against that, because I think there's an example where, for example, pathogens can dominate ecological environments where there's little competition. So if you take, for example, the stomach, it's an extreme niche, I know, but low pH, very low microbial diversity. But a few adapted microbes survive there, and we have Helicobacter pylori, a pathogen that damages the stomach wall, etc. So does that not argue against your proposition?
I don't think so, because some, like Helicobacter pylori, I know that it can live in this very acidic pH, but of course there are many factors that affect why this bacteria grows, such as the immune system that they have in the body, and also the proportion of the good bacteria. The probiotics that you have in your body also will compete with these pathogens and make these pathogens will not grow as high number as the good bacteria.
But in the stomach, there are very few bacteria that can survive in that harsh environment, and the food digestion has really only just started.
Yeah, it's true, but it really depends on which part of the food, which part of the stomach, right? I mean, like the stomach is an exception because you have very extreme pH on that. But what I mean in here is like the overall health. It doesn't mean only the stomach, but also, for instance, the intestine, the colon, and so on. That's, yeah.
I think we can agree. That is an extreme niche, and I understand your point, and I think we can agree that there is microbial culture or competition, which is dependent on the diet. And
Yeah.
I think we can agree that's a situation that we both have accord with. Yeah. I'd like to move on now and ask you a question about your choice of efflux pumps. So this is a very important strategy, of course, and you chose for gram-positive SNOR-A. So I'd like to start by asking you how conserved is the NOR-A sequence among gram-positives? Do you have a rough estimate of that?
Highly esteemed opponent, thank you very much for your question. So your question, how conserved is NOR-A efflux pump and why we choose this NOR-A efflux pumps as our target for gram-positive? Yeah. Actually, this is interesting because what we found with the Staphylococcus aureus, this NOR-A is overexpressed in 43% of MRSA strain. And interestingly, fluoroquinolones is considered as broad-spectrum antibiotic, but in this strain that we are doing the research, it has resistance against fluoroquinolones. And based on the previous study from Katz's group, they found that the resistance in this strain was because they overexpressed the NOR-A efflux pump. However, I agree that Staphylococcus aureus also have other type of efflux pumps, and because this NOR-A Staphylococcus aureus has been studied worldwide, and this is a good model to study the efflux pump, that's why we started with the Staphylococcus aureus, and we studied NOR-A efflux pump.
I understand better your choice now, considering Staph aureus and resistance, etc. But let me end this particular question with, given the inhibitors you have against NOR-A, if you want to use them more broadly against other other pathogenic bacteria, would you have to start again, or could you use NORA as a scaffold? And what's your idea about that? How likely is it that would inhibit other bacteria? Have you tested other bacteria, gram-positive bacteria?
Your question is if we find the inhibitors that targeting this NORA, is this possible that this compound also inhibit other pumps?
Inhibit other gram-positive efflux pumps, yes.
There is possibility of that. Because in Staphylococcus aureus, NORA is one of the multi-MFS superfamily transporter, but it also has other efflux pumps which belong to the same family of transporters, such as NORB and NORC. And this efflux pump also have similarities with NORA efflux pumps. So it is possible that if our compounds inhibit the NORA efflux pump, they can also inhibit other efflux pumps which are in the same MFS family.
Respected candidate, I see my time is up, so I had better pass.
Oh, you would have some more time.
I have some more time.
Yeah.
Oh, okay. Yeah, and I have a few technical questions about the selection of the inhibitors. So I'd like to first of all ask about the possible effect on the proton motive force of the compounds that you're adding. So, because if they permeabilize the membrane a little, you might lose proton motive force, which is a driver of ATP production. So did you test any membrane energetics?
Yeah, actually with our prenylated isoflavonoids, because those compounds also have antimicrobial activities. So we also try to test actually whether they are active as proton motive force inhibitors. However, during the setting up of the method, we found that these compounds can have fluorescence quenching effect with the dye to test the proton motive force inhibitor effect. So this is something that we do not know yet, but for the epi activity, we make sure that the concentration that we tested was below the MIC value. And we also answered that there is no membrane permeabilization effect at the tested epi concentration. So that's when we test at epi concentration, we answered that there is no membrane permeabilization effect and there is no antimicrobial activity at that concentration.
And did you always strictly test the pH in these assays? Because that could also affect the proton motive force, the external pH.
Yeah. During the experiment, we work on the physiological pH because we work with PBS buffer and it has pH 7.4, so we didn't test with different pH for the experiments. Yeah.
Thank you very much.
Okay, thank you very much, Professor Wells. The discussion will be continued by Dr. Hartkorn, who is Research Director Microbiology and Antibiotic Discovery at the Institut Pasteur in Lille, France. Dr. Hartkorn, welcome. The floor is yours.
Thank you. Dear respected candidate, thank you for providing us with this thesis. I enjoyed reading it, and I have a number of questions, as you will expect. First of all, I would like to look at Chapter 2, your first scientific chapter, and look at your ethidium bromide uptake assay. And I was wondering first, why did you it's an uptake assay, so you often look at the rate of uptake. So why did you decide to look at a endpoint uptake as a measure of your fold change rather than the rate of uptake?
Highly esteemed opponent, thank you very much for your question and your kind words. When we did this experiment, we tried to look for the most doable experiment. So it would be nice to have the kinetics of this ethidium uptake, but for the screening, for the reason of the efficiency, when we want to assess whether there is an efflux pump inhibition activity, we think that ethidium accumulation assay is enough to see whether there is an inhibition over the pumps or not. And then for another confirmation, we also use the checkable assay to see whether there is a potentiation with antibiotics, which further support the results of our ethidium accumulation assay.
Okay, thank you. So you're looking at inhibition of an antibiotic efflux pump, and you decide to look at ethidium bromide rather than an antibiotic. Can you explain why you took that decision for the screening? So your screening was done with Ethereum bromide, not with an antibiotic.
Yeah. This is because Ethereum is the substrate of the efflux pumps, and these experiments is quite fast to do. So what we expect, because Ethereum, when they intercalate with DNA, they can give the increase on fluorescence. So this can be an indication if the efflux pumps are inhibited, then more Ethereum will accumulate inside the bacteria. It can intercalate with DNA, and then there's an increase in fluorescence. Well, for the antibiotic, what we see is the potentiation effect. Okay, how many fold change of potentiation that we think, and then not all of antibiotics is the substrate of certain efflux pumps, but Ethereum is the substrate of most of the efflux pumps. So it is more practical to do with Ethereum, to start with Ethereum and then with the checkerboard assay.
Okay, that is a choice. Going against that, you have some conflict between the results of your Ethereum bromide uptake and your antibiotic boosting. So if I may, as you correctly pointed out in your document, you show that in the Ethereum bromide uptake, there is hyper accumulation of Ethereum bromide even in the absence of NOR-A, in the NOR-A knockout. And you note that there might be some additional activity. But you show full potentiation, let's say. Now, if you look in your table 2.2, which I think is page 53, we're looking at the activity of ciprofloxacin in your NOR-A overexpressing strain, which, if I can read correctly, is 8 microgram per ml, and in your NOR-A mutant is 0.25. Is that correct? So there is a large difference between the absence of NOR-A and the overexpression of NOR-A on the MIC of ciprofloxacin.
Could you repeat again your question? For which compound?
Ciprofloxacin, the fluoroquinolone.
Yeah.
So there is a large difference if you genetically knock out NOR-A, so when it's completely no NOR-A, versus when it's overexpressed. I think it's 32-fold, if I calculate correctly. Now, if you then look at the boosting from your molecules that caused hyper-accumulation of your Ethereum bromide, it only caused a small dent in this difference. It only 4-fold. So can you give me your interpretation of the differences in these two messages? So one message, Ethereum bromide, hyperaccumulation, looks like full inhibition, and in the other ciprofloxacin assay, you have only a partial reversion of the efflux.
Yeah. So for the Ethereum accumulation assay, well, this is really, there are many factors that affect the fold change, actually. Based on my experience, when we did this Ethereum accumulation assay, well, it showed the increase in fluorescence over the time in 1199B, but the values of how many increase the fold change itself, it's sometimes different upon different experiments. And I would say Ethereum accumulation assay is mostly can be used as qualitative parameter to see whether yes or no there is an efflux pump inhibition. And then the antibiotic potentiation is more on a quantitative way to see, okay, how many fold change that you see when you combine your compound with the antibiotics, which is known as the substrate for the efflux pump. So that's the different.
Okay, so if you were to go back in time with your current knowledge and replan the screening of your libraries of extracts and molecules and very interesting structures, would you change anything, or would you use the same approach like you did in?
I would start from Ethereum accumulation assay because it's easy to see whether there is an efflux pump inhibition or not. And after I see an indication that the compound inhibited the efflux pumps, then we can move forward to the antibiotic potentiation. This is because for the antibiotic potentiation, we also need to check what kind of substrates for the efflux pump that we have. And also, There are many concentrations that we want to test when we do the checkerboard assay, but when we did the ethidium accumulation assay, we just can start the screening with small concentration and see whether there is an efflux pump inhibition or not. So
you
it is
believe-
more handy.
You believe, respected candidate, you believe that it was a good way of predicting good efflux pump inhibitors, the ethidium bromide screen?
It is good, but it is not enough. We need both of them together. So we need the ethidium accumulation together with antibiotic potentiation, the checkerboard assay.
Okay, excellent. So I have a question about the membrane permeability assay. And I agree it's a very important assay to do because compounds can always have off targets. My question is, do you believe the assay you chose, we have to make choices, maybe you can explain the choice that you made. Do you believe this assay is able to show you all types of membrane permeabilization, or because you stated very clearly in the question before that there is no membrane permeabilization? So can you be that strong in your statement?
I stated there is no membrane permeabilization based on PI abscess assay, based on the propydium iodide. If it's, but I cannot guarantee that the permeabilization do not occur. We need another experiments to check about that. For instance, we can use another dye, which is more sensitive to assess the propydium, to assess the membrane permeabilization effect. We can also use a fluorescence microscopy method to check whether there is a membrane permeabilization effect. Based on the propydium iodide uptake, there is no membrane permeabilization, but it doesn't guarantee that the small permeabilization occur.
Did you consider, and perhaps you have done, doing experiments on antibiotics, boosting of antibiotics, potentiation of antibiotics that are not substrates of NORA, and evaluate whether they are also potentiated or not because of potential membrane permeability?
That's what we did, actually. With the NORA, with prenylatives of flavonoids, we also test the activity of erythromycin, because erythromycin is not reported as substrate for NORA in the NORA overexpression strain. And we didn't see the potentiation with the erythromycin.
That was excellent.
Yeah.
I found it a shame that you could not do it on the NORA mutant because I think you had a resistance cassette, right?
Yes.
And so it would have been nice to also have seen another antibiotic perhaps where you
Yeah.
could have tested in them both.
Yeah.
Okay, excellent. How much time do I still have?
Short question can do. Yeah.
Okay. My question is more in general. Now you have identified in your 3 chapters a number of potential candidates to develop. Can you give some thoughts on which steps you would envisage would be needed to get them to progress further towards a antibiotic or a booster of antibiotics in clinic?
In our study for the NORA efflux pump inhibitor, we didn't use various mutants like we did with E. coli study. And for this one, we also, if I have time, then I would like to test in a clean NORA upregulated strain. So basically the Staphylococcus aureus strain, we knock out the NORA, and then we introduce another NORA, and then we also have this, like we did with E. coli, we have ECO35, which this strain has knockout of 35 efflux pumps, and we only introduced our gene of interest on that. And then, so we know whether the compound's activity is mainly due to the NORA inhibition or not. And also, another study that we miss here is the binding affinity, because with the NORA we only work with Staphylococcus aureus overexpression, but we do not know the binding affinity of these compounds against the NORA protein itself. So that's also something that we miss before we optimize further the compounds, because we have no idea what binding location that this compound has in NORA.
Thank you.
Okay, thank you very much, Dr. Hartkorn. The discussion will be continued by Professor Van Westen, who is a professor of AI, Artificial Intelligence and Drug Discovery at the Leiden University. Professor Van Westen, welcome as well to you, and the floor is yours.
Thank you very much, Mr. Rector. Respected candidate, firstly, I would like to convey my appreciation on the completion of your thesis. It is a nice piece of work, and I'm charmed by the coupling of experimental and data analysis, and I should like to extend that congratulations also to your promoter and co-promoters. However, I do have some questions, and as was kindly introduced, my background is more in cheminformatics and machine learning, so I would also like to focus my questions a bit there, but before we go there, I have two generic questions that I would like to ask you. Firstly, your work focuses on efflux pump inhibition, and you have very nicely mentioned why, how, and the choice of assays. But I was wondering, if you inhibit efflux pumps, I mean, it's not the only mechanism of resistance. Do you expect other forms of resistance to occur or increase?
Highly esteemed opponent, thank you very much for your kind words and your questions. Your question, if we inhibit efflux pumps, does it mean that there is no other resistance mechanism occur? Well, I cannot guarantee about that. But we know so far that the efflux pumps are involved in the pump outs or extrusion of various antibiotics, which are necessary like for the infections or to kill the bacteria. And by knowing that the efflux pumps, they are very important and they're involved in the resistance mechanism, so we aim to target these efflux pumps to increase the potency of antibiotics, but it doesn't guarantee that when we inhibit the efflux pump, there is no other resistance mechanism will occur.
And so I'm always charmed by evolution, so can you speculate on one or two mechanisms that may occur?
The resistance mechanism?
Yes.
If I may speculate, could be the adaptive resistance mechanism will occur because the bacteria was not used to this efflux pump inhibitor before, but upon the exposure of efflux pumps, by the time it will get adapted with that and it try to avoid these efflux pump inhibitors. That could be possible.
Thank you. This is also the direction I wanted to go.
Yeah.
And because in the end, of course, you want to kill bacteria, but you also want to treat patients. So are there, to your knowledge, mammalian equivalents of these efflux pumps that may also result in side effects or toxicity?
Yeah, that's possible. So when we found the compounds that potentially active as efflux pump inhibitors in bacteria, we also need to ensure whether the compounds, they are also acting against efflux pumps in humans, such as PGP protein, for instance, the ABC transporter in human. They're important for the cytotoxic, for the cancer therapy. So it will also affect, if the compounds inhibit the PGP protein, it may also can cause this resistance over cancer drugs. And that's also the next study. Actually, when we have time, that would be nice to predict the activity of this compound against the transporter protein in humans.
Do you think there will be an effect?
I cannot say it right now, but these two efflux pumps, the efflux pump in bacteria, the RNA efflux pump, and MFS transporter, so they are different type of efflux pump compared to PGP, and they are not similar efflux pumps. So that could be the compounds do not inhibit the PGP, but of course we didn't prove it, so we do not know yet, but there is possibility.
Yeah, I'm in favor of time. I would like to move more to my own core expertise, which I prefer. And I should like to thank my predecessors because they've paved the way for some of the questions I have. When you did a QSAR in chapter 3, which was really nice to see also because even with relatively little compounds, you were able to extract meaningful correlations. But that always makes me wonder about how you got there. For instance, standardization. Can you elaborate on your standardization procedure?
So when we did this QSAR study, it's also very challenging because the compounds that we have is no more than 50 compounds. So the first problem that we had was like, okay, what kind of parameter that we can use to build this QSAR model? And it's so difficult because we don't have information with the kinetics, and the data that we have now is less variability. For the standardization, actually, we didn't really do this standardization because we just used the experiments that we have with Ethidium accumulation assay and also the antibiotic potentiation assay. So we only assessed the fold change and also the potentiation effect, how many fold reductions in antibiotics MIC. So that's why in the end we come up with this QSAR binary instead of the multiple linear regressions model because, with the standardization, this is something that we can, optimize later.
So you took it also as input the smiles from PubChem, right?
Yes.
And I'd like to take you to page 82, where you also mentioned that you do partial charge correction followed by energy minimization using the MMFF94X force field.
Yeah.
So I was wondering, did you consider the pH at which you do this?
No, we didn't consider the pH.
Can
so,
you estimate what it's probably going to be?
So that could be that the compound's not in the.
Well, I'm going to help you a bit here, but it's going to be probably 7.4, which is in line with your
Yes.
experiments.
Yes.
So I would say that's good. Is this also the pH at which you expect the compounds to be active
Yeah.
on site?
Yeah. Because we use this physiological pH when we work with bacteria, and it's in 7.4.
Yeah.
So that's why that's what we expect.
And when you then go to the end of your QSARs, you describe them very well. And then at page 92, you have two binary models that you describe. And one of the QSARs, they have as main contributing descriptors PEOE VSA FF POS plus VSERV HL2. And the other has PEOE VSA FP NEC and log P. So to me, that seems that quite similar properties come out, right? They're quite similar, and they're both related to lipophilicity. Have you considered trying a model only on log P to see if that has an effect? And that relates also to the standardization that you were doing.
Can you repeat again your question?
So have you tried doing a QSAR with only log P?
No. No, I didn't consider to do with the log P because what I mean, because log P is not the activity of the compounds itself. So that's why we didn't consider the use of log P for the QSAR model. So we only consider about how we assess the activity of the compounds. And so we use the most commonly used to assess the activity.
Your fingerprints, your descriptors, they are both fractional polar surface van der Waals based, right? So they have a relation to the van der Waals area. But the one is positive and the other is negative.
Yeah.
Do you have any idea why that is?
Yeah, this is also an interesting question. But this is something that, so this is something that actually expected because the first descriptor was obtained from the ethidium accumulation assay, and the second descriptor was obtained from the antibiotic potentiation assay, which means that this negatively charged also might be linked to the antimicrobial activity because how we assess the antibiotic potentiation, right? But this is not the case for ethidium accumulation assay because the negative charge is not that important for the ethidium accumulation activity, but this is more important for the antimicrobial activity. The intersection between this antimicrobial, the epi activity, could be,
Yeah, and you have 160 descriptors that you tried. Have you tried similar descriptors that also have partial charges, positive or negative, in that pool?
No, we didn't try with that.
So you're quite confident that they should work, but it could be that there are other sub-descriptors that also would show an effect.
That is possible, but what MOI did, because we use a software which is called MOI, so they did the iterations, and these two descriptors that appear here is the most correlated one. So the descriptor that are not highly correlated, they just remove it, and then it's not appear in the model. So that's why we end up with these two descriptors, because this is the two most correlated descriptors based on the iteration.
I'm going to cut you a bit short here, because can I ask you, respected candidate, if you can ask one of your paronyms to read proposition 4, please?
Their paranym, could you please read proposition number 4?
Progress in drug discovery is slowly reducing the use of traditional herbal medicine.
Yeah, thank you. When I was reading this proposition, I was thinking, wow, I agree. So, would you argue that we would need to bring back more pharmacognosy departments or not?
Yeah, in this proposition, I want to explain the context first. The traditional herbal medicine that I mean here is the use of plants or herbs based on their traditional knowledge and based on their cultural beliefs. So the isolation of compound from plants, for me, it's still considered as modern medicine because you need to isolate the compounds and then you need to optimize the compounds, becomes a drug. So that's what I see here is like in some parts of the world, especially in developed countries, people tend to use or prefer to use the modern medicine because they have more information about their efficacy, about the side effects, so they have more confidence in using that medication instead of using traditional herbal medicine. That's how the preposition comes from.
Would you argue or would you agree with me that actually that can be a source of inspiration that we as Western medicine are not considering sufficiently?
Well, I would say that, yeah, we are still considering the phytochemicals or pharmacognosy, but well, we are as a medicinal chemist, for instance, we just use this kind of plants in more efficient way, because if you use the traditional herbal medicine, you just use these plants or these certain herbs, and then you do not know how much kilogram that you want to use, right, to treat a disease. But then as a researcher, they try to isolate the compounds, and they know how many micromolar of the compounds or milligrams of drugs in the end that can give the efficacy.
I think I tend to agree with you, and I would also argue that by isolating, we sometimes miss something that can be present because of a combination of different active substances. And with that, I would like to give the word back, because I think I'm on time.
Thank you very much, Professor Weston. The last opponent is Dr. Liu, who is assistant professor here at Food Microbiology. Dr. Liu, also welcome to you, and the floor is yours.
Thank you, Mr. Chairman. Dear respected candidate, first, also my congratulations on the completion of your thesis. I read with great interest. The book looks very beautiful, and I really like your cover. Look, so this is in nature. We have this little path, which illustrates actually the bacterial membrane, and it holds more than one secret to the survival of bacteria, because you have the efflux pump, and the membrane itself is a barrier between life and death as well. Related to that, I noted multiple times in your thesis that you really emphasize for the criteria to define EPIs, there should really be absence of membrane permeabilization effect with the effective concentration of the EPI. I do understand that's required for a very neat definition. But now, if you are just looking for a compound that is really working well against pathogens, can I argue that if a compound that has a dual function, that it has both the epi activity and membrane permeabilization effect, would it be more favorable?
Highly esteemed opponent, thank you very much for your kind words and your questions. Yeah, this is also something that I'm still thinking, whether it is nice to have compounds that can have the epi activity and also membrane permeabilization effect as the antimicrobial mechanism, for instance. So they can have this to effect, and then it is more efficient to kill the bacteria. But, well, this is also something interesting to think about because it is more efficient. However, we also need to consider, because if the compound have membrane permeabilization effect, they can have this bactericidal activity. And when the compound has this bactericidal activity, there is higher chance that the bacteria recognize this compound as harmful compounds, and then in the end they can develop resistance like the same mechanism they develop with the antibiotics. So that's something that also need to be taken into account. But yeah.
I see your point there, and following your discussion just with Professor van Westen, you also did mention the epi could also lead to resistance development. Can we imagine that if I have a dual function of killing, it's more difficult for the bacteria to develop resistance?
But I'm not sure actually with that, because if the bacteria, if the compound can have this dual mechanism of actions, then it is more harmful for the bacteria, as I explained before, and it is more bactericidal. And could be the bacteria can develop the resistance mechanism faster rather than using the compounds that mainly only target the efflux pumps.
What doesn't kill them makes them stronger, right?
Yeah.
I see your point. All right. And as we spoke about the antimicrobial resistance development, I think it is common sense that you need to apply an effective drug with sufficient concentration and treatment for sufficient time. Otherwise, you are leaving more room actually for them to develop resistance or to survive. So that brings me to my questions about the effective concentrations of the EPIs. In this study, you have been studying the effect at 37 degrees and in the standard microbial cultivation media. And then in chapter 5, you proposed some applications of the EPIs, maybe in food preservation. And you mentioned that food matrix, pH, and temperature can all influence the activity of the compound. I can imagine you would need to test for all this, but do you think, with your knowledge, is it possible to guess what would the effect of, for example, a lower temperature be on the effectivity?
Yeah. If I may guess, lower temperature could maybe decrease the activity of the compounds.
Why?
Because the bacteria, they are not in active pumps during that state. For preservation, we use this low temperature. That could be, yeah, the compounds, they are not actively, maybe they bind to these efflux pumps, but I don't know, because I hypothesized in my thesis that they could have this competitive mechanism. So it means that this compound competes with the antibiotics or the preservative compounds, and then they can, instead of the efflux pump, instead of the antibiotic that are pumped out, the compounds itself can be pumped out. That's also the possible mechanism that we hypothesize here. But then when the bacteria, they're in the preservations, I mean, like the food, they're in the preserved condition, they have low temperature, and the bacteria is not active. I'm not sure about this competitive mechanism, whether they occur in that state.
Mm-hmm. So if the bacteria is not very metabolically active, you expect less effective for the EPIs as well.
That's what I'm thinking now, but
Yeah.
I have to test if I want to know further about that.
That's always the case. And what would it mean for example, the hydrophobicity interaction for your compounds if temperature is lower?
When the temperature is lower, I don't think that it will affect the hydrophobicity.
Okay.
Because I think what the hydrophobicity of the compound will affect the membrane more, and then when temperature is lower, I don't think that membrane condition will change.
Okay. And then with the food matrix, I can imagine whether there is a lot of proteins or amino acids swimming around, they can also interact with your EPI components. So I'm wondering, is it fair to say that when you talk about an effective concentration of the EPI, you can only say it for the exact condition you have been testing it for?
Yes, it's true. That's also what I realized, because in every experiments that I did, there is a different optimum concentration to see the effect. And yeah, but that's why we try to make a threshold. We try to make, okay, we can assess the compound as EPI when they show the effect at sub-inhibitory concentration. That's what also we propose. It should be at least at 1 for MIC. Yeah, but I can see the difference among different experiments.
And related to that, I want to link this definition of the EPI effective concentration to when you check them with the cytotoxicity test in the human cell model. Mm-hmm. I looked up in chapter 2 that I think the human cell model was cultivated in DMAM with fetal bovine serum, while your bacteria was in a different media, of course. Was it completely fair to compare the EPI concentration and refer cytotoxicity in a different culture environment?
Well, the cytotoxicity effect is that's for a different purpose, because when we did this cytotoxicity effect, we we didn't do the proton multiforce inhibitor. And then when we check when the compounds is proton multiforce, they can have side effect where they have the high cytotoxicity. So the aim actually for this cytotoxicity assay, we want to know how the compounds behave toward intestine cells. So that's why we use this CACO2 cell line. So it's in different purpose with the experiments with the bacteria.
I think I was more referring to the matrix effect. Like if your two different media are different proteins or concentrations are present, they may already interfere or deactivate part of your molecule numbers, but we can leave that for now. And now I would like to discuss a bit more about the applications of the EPIs you proposed in Chapter 5. And for that, could you ask one of your paronyms to read out loud proposition number 3, please?
Dear paronym, could you please read proposition number 3?
The increase in antimicrobial resistance is a consequence of non-existent policies.
Thank you. If I understood correctly, you're trying to say that if there's not enough regulation, it leads to maybe use of antimicrobials and lead to resistance, if I'm understanding correctly.
Yeah. The context in these propositions, the policy that I mean is uniform international policy on the antibiotic use for non-medication treatment. Since 2006, we know that the EU banned the use of antibiotics for as a feed additive, for instance, in animal farming. But I realized that this is not the case for other countries in the world. Like, for instance, those countries with low middle income countries, there is no strict rule on the antibiotic use for non-medication purpose. So that's how this proposition comes from. So I don't see this similarity for the regulations among all the countries.
Yes, thank you. And now, in that view, if I look at your the application you proposed for the EPIs, and sometimes in combination with antimicrobial phytochemicals, you mentioned in figure 5.7, that sometimes you say they can be used as a prophylactic treatment for animal feed or disinfectant against the foodborne pathogens. So what kind of policy do you think should be in place to make sure that this kind of application is not stimulating resistance development?
Yeah. What in this proposal, like to use these phytochemicals that are acting as antimicrobials together with EPI, might be solutions for this kind of resistance. But for what kind of policy, I would say that there is more research to do about that. With the antibiotic, we know that antibiotic has been known as resistance, or it induces resistance in a bacteria. But with the compounds itself, we do not know yet whether these compounds can develop resistance.
Thank you also, Dr. Liu, for your opposition. And now I join the meeting, and the examining committee will withdraw for consultation. Please be seated. I hereby reopen the meeting. The Academic Board of Wageningen University, represented by the Deputy Rector Magnificus and 7 committee members appointed by the Academic Board, having noted the content of a thesis entitled Exploring Phytochemicals as Efflux Pump Inhibitors Against Multidrug Resistant Bacteria, with propositions, having heard the defense of that thesis, has decided to confer the degree of doctor on Marina Irianti, born in Blitar, Indonesia, on July the 22nd, 1992, and to grant to her all rights and privileges ensuing from that doctorate by law and custom. The Academic Board assumes that you accept your duty as a scientist to execute your future research ethically and with due diligence, according to the Netherlands Code of Conduct for Research Integrity. I now invite the promoter, Professor Dr. Vinken, to present the new doctor with the degree.
You have heard the decision of the Academic Board of Wageningen University to confer on you, Marina Irianti, the degree of doctor. It's now my honor to present you with the degree, signed by the Deputy Rector Magnificus, the promoter, and the 2 co-promoters, and sealed with the Great Seal of Wageningen University. I first invite you to sign the degree as well. With this signature, you declare to act according to the Netherlands Code of Conduct for Research Integrity in the future.
You may rise and wait here, please. No, I will put this on the tables first. You can wait here.
Allow me, Mr. Deputy Rector Magnificus, to offer my congratulations and to add a personal address. Dear Dr. Irianti, dear Marina, it's my pleasure to be the first one to congratulate you with obtaining the degree of doctor, also on behalf of your co-promoters, Dr. Carla Araya-Cloutier and Prof. Martin Pos. In my congratulations, I would also like to include your husband, Siddiq, your family that came over all the way from Indonesia, friends and colleagues as well. Studying phytochemicals as bacterial efflux pump inhibitors was a new direction for us at food chemistry when you started your PhD in 2020. A few years earlier, Carla had investigated the antimicrobial properties of prenulated isoflavonoids from Fabaceae against gram-negative bacteria and found that efflux pumps played a crucial role in resistance of these natural compounds. Building on that insight, your project aimed to identify efflux pump inhibitors from plants, so-called phytoepis, to be used in combination with plant-derived antimicrobials to help overcome bacterial resistance mechanisms. We were particularly pleased to collaborate with Prof. Martin Pos, who is here on the podium, from Goethe University in Frankfurt, and Prof. Paolo Ruggerone and Dr. Giuliano Malocci from University of Cagliari in Sardinia, Italy, all renowned experts in the field of bacterial efflux pump systems. I would like to thank them for their support and constructive contributions throughout your work. In your research, you explored phytoepis from edible plant species targeting efflux pumps in multi-drug-resistant pathogens, Staphylococcus aureus and Escherichia coli. You demonstrated that prenulated isoflavonoids can exert a dual mode of action in S. aureus, functioning both as effective epis and as antimicrobial agents. You also identified a new class of interesting phytochemicals, indole alkaloids from Brassicaceae, as promising epis against E. coli. Beyond experimental characterization, you integrated in silico methods to better understand the structure-function relationships and mechanisms of action of these phytochemicals. Your first two experimental chapters have been published in Q1 journals, and the third one is currently under review. Mr. Deputy Rector, allow me to give the floor to Dr. Araya Luce to address some personal words to the young doctor.
Dear Marina, congratulations on your degree of doctor. Back in 2019, I was fortunate enough to receive a Veni grant to explore a new research line on plant-derived efflux pump inhibitors to combat resistant bacteria. For this project, I wanted someone with a background in biology and pharmaceutical sciences. Luckily, after completing your medical pharmaceutical studies in Groningen and working at the University of Indonesia researching medicinal plants, you were longing to return to the Netherlands. Beyond the amazing weather in Groningen, I suspect you also miss your husband, Siddiq, who was still living here at that time. This project was the perfect opportunity for both of you to reunite. When you started your PhD project, you quickly immersed yourself in the most up-to-date literature on efflux pump systems and inhibitors. You read all the top papers, learned the names of all the experts in the field, and began designing an impressively ambitious set of experiments. We appreciated your enthusiasm, although we had to bring you back to earth a few times. Attempting to combine chemical, microbiological, structural, and modeling experiments into one single study might have been a bit too ambitious. After aligning ambitions with practical realities, you also had to adapt to the life in the food chemistry department. Understanding structure-function relationships and chemical classifications was new for you. Talking about chemical classes and molecular properties wasn't, and I still suspect it still isn't, exactly your cup of tea. On top of that, you had to adjust to supervising students here. You had experience from Indonesia, of course, but working styles and schedules are a bit different in the Netherlands. I still remember having to mediate between you and your first student, who wasn't too thrilled when you asked her to finish one last experiment after 5 o'clock in the afternoon. To be frank, we supervisors also had to adapt. Your strong opinions often made our meetings lively and interesting. Perhaps your pre-discussions with your colleague and now paranymph, Sylvia Kali, who proudly called herself the recalcitrant student, assisted in sharpening your arguments before our battles, meetings. In 2023, you spent 6 months in Martin's lab for a research secondment. There you performed quite a range of experiments, from protein ligand binding assays, plate dilutions test, to protein purification and co-crystallization. Despite our careful planning, we overlooked the detail that you were technically moving to Germany and that immigration authorities might want to know about it. Working kind of illegally and trafficking white powder, your compounds, across the border was risky in retrospect. Thankfully, no immigration officers came knocking on your door, and you're still here with us. I'd like to take this moment to sincerely thank Martin and everyone from the lab who welcomed you and supported you during that time in Germany. Outside the lab, you truly enjoy your stay in Frankfurt. You're a city girl, and Wageningen was starting to feel a bit too small. I could tell that you and CET enjoy your secondment in such a vibrant international city. With food being one of your passions, Frankfurt was the perfect place for culinary exploration. You made it almost a personal goal to visit as many restaurants as nationalities in Martin's lab. Not sure if this goal was completed, but I still remember the delicious Korean barbecue restaurant you took me during my visit there. After returning to food chemistry, it was time to dive into some in silico work. Fortunately, we had great support from Paolo and Giuliano in Cagliari, who assisted with the molecular docking experiments of ACRB and our candidate EPIs. Later, you went on to perform a quantitative structure-activity relationship study. Not easy at all, as building QSAR models turned out to be far less straightforward than you first thought. Marina, you were also involved in different activities here at Food Chemistry, helping to supervise BioFIF, AVAF courses, serving on the PhD trip, organizing committees, volunteering to cross borders to collect essential suitcases, and even organizing Jean-Paul's visit to Yogyakarta in Indonesia. Outside of work, you were just as active, hosting dinner parties nearly every weekend while building a big group of friends all across the Netherlands, some of them present here. Marina, I want to thank you for your hard work and your dedication throughout this journey. I'm truly proud to see you here today, holding your PhD diploma. Congratulations once again to you, to your husband Siddiq, your parents, your brother that are coming from Indonesia, and I wish you a very happy and memorable day. With that, I return the floor to the Deputy Rector.
Thank you very much, Dr. Area Loutier. Dear esteemed Dr. Irianti, this marked the end of this ceremony in which you successfully defended your thesis in public. On behalf of Wageningen University, I would like to congratulate you on obtaining your degree. I would also like to extend my congratulations, it has been done before, to your husband, to your family sitting here in front, and to everybody here present, and maybe also watching from Indonesia by the internet. You really worked on a very interesting topic. We share that interest in the interface between natural compounds and medicines. And also hearing your defense, I agree with one of your opponents that I think also pharmacognosia should come back. It also makes it a topic which is very well fitting in the vision and the mission of Wageningen to explore the potential of nature to improve the quality of life. As I understood, you don't have a job yet, but you are looking for something in academia. We wish you all the best with that. And at least we also hope that you will continue to be a Wageningen ambassador. I would also like to thank you as members of the committee for being here today, and also for all the evaluations you did. So Professor Wells, Dr. Hartkoren, Professor Van Wester, Dr. Liu, thank you very much. Your contribution is really appreciated. Without your contribution, it would be impossible to maintain the quality of our thesis. And finally, I would like to thank you as audience for being here on this Friday afternoon, attending the ceremony and showing your appreciation for the work of our young doctor, new doctor. So with this, I close the meeting.